Well, welcome everybody to our first seminar for the spring quarter, GBSBC, Genetics and the Bioinformatics and Systems Biology. So just to kind of give our speaker a sense of who we have in the audience, raise your hand if you consider yourselves a genetics person. Okay, raise your hand if you consider yourselves a bioinformatics and systems biology person. And you can raise both hands if you want. Great, so that gives you a little bit of a sense of who's here. Some of our BISB program students have not yet met our brand new graduate coordinator. This is Amber, and Amber is going to be our problem solver for all problems. And Amber and I are already working together on pretty much everything that happens in BISB. So welcome, and I'm going to ask our student host to introduce our speaker.
Did you tell them about pizza? The bad news about pizza?
Yeah.
I can do it real quick.
Okay, there you go.
So yeah,
So
I gotta be the bearer of bad news. So pizza is gonna be coming somewhere in between the talk. So what we're gonna do is just have you get it on the way out. So try not to disturb the talk or anything like that. Sorry about that. Okay, so I'm gonna go ahead and introduce Professor Soo-In Lee, who has courteously joined us from the University of Washington in beautiful Seattle. Professor Lee is a Paul G. Allen Professor of Computer Science at UW. She earned her PhD at Stanford University under the mentorship of Daphne Koller. She joined UW in 2010 after serving as an assistant professor at the Computational Biology Department at Carnegie Mellon University School of Computer Science. She's been recognized for a lot of her groundbreaking contributions to the fields of AI and biomedicine, and has received several accolades, including the National Science Foundation Career Award, the International Society for Computational Biology Innovator Award, and a designation as an American Cancer Society Research Scholar, among others. And like I said, she's made very recent contributions surrounding the fields of especially explainable artificial intelligence in biomedicine, including, I'm guessing, especially the PhD students here have used the SHAP framework probably at some place if you've done any kind of machine learning before. So we're really happy to have Professor Lee here to give a talk. And with that, the floor is yours.
Thank you so much, Adam, for such. Thank you, everyone. Okay, so today I'll be presenting the research from my lab on the topic of explainable AI. This is probably the word you will hear the most frequently today. It refers to a subfield of AI aimed at enhancing the interpretability of complex machine learning models. And then I will focus, this talk will focus on the field of biomedical sciences. Okay, all right. So before we get there, the work I want to say, the work I'm about to present were primarily carried out by my students in the Ames lab. We consist of passionate researchers pursuing their PhD in computer science at the UW, including the individuals from the MSTP program, it's MD PhD, concurrent degree program, and then still, pursuing their PhD portion in computer science. Okay, so I indicated with different background colors, and then we work on a broad spectrum of problems. You will hear my talk will span all three fields. So one is developing explainable AI techniques, AI and ML area. And then B, basic biology. So the theme being identifying causes and treatment of challenging diseases. My lab is focusing on currently cancer and Alzheimer's disease. And then C, developing and auditing clinical AI models. Okay. So this talk will consist of three parts. In the first part, I'm going to give you many high-level examples. You will basically see a slide or two per project. But the purpose is to give you some high-level message in terms of how explainable AI can address new scientific questions in biomedicine, and then with the primary focus on understanding neurodegenerative diseases, and then also biological age. And in the second part of the talk, we will explore how explainable AI can pave the way for new research directions. So you want to hear about how we go beyond the just interpreting model and then do more than that. This will, give some insights into, how explainable AI needs to evolve and then improve to effectively address a broad spectrum of real-world problems in the biomedical field. Okay, so in this talk, as I said, we won't delve into, deeply into a couple of projects. Instead, we will explore numerous examples, many examples of how explainable AI can revolutionize potentially biology and medicine, and then that way I will, convey high-level message about, transformative potential of XAI, and then in fact the methods we are developing in my lab, they are general. They are universally applicable to broad spectrum of a problem. Even methods developed for, say, single-cell, transcriptomic data analysis, it can be applied to the analysis of clinical data as well. So please, focus, because some of the methods you will hear about, you may apply to your own research, okay? And then you can have some interesting results. Okay, so now, the key goal of explainable, I kept telling you explainable AI without even really, defining it. Okay? So the key goal is to try to understand why a certain output was generated by a black box machine learning model. So, let's say you want to predict dementia onset, imminent dementia, say after 3 years, whether this person is going to have dementia or not, based on a set of features, consisting of, say, demographic features and clinical data, some genetic information, and also extensive, the cognitive exam results. Okay, so you have this high-dimensional data. You take that as an input. You use high-performance, model that's going to give you high prediction accuracies. But this a traditional machine learning paradigm. You can't really answer natural questions in terms of how this prediction was generated, what drove this prediction. Okay, machine learning model is completely a black box. It's because, with a large modern data set, you normally have a large sample size in training data. The best accuracy is achieved often by complex models, which even experts struggle to interpret. So the idea of explainable AI then is to explain the output of the black box machine learning model based on the contributions of each individual features for that particular prediction. So now you're focusing on one individual, one sample, one prediction, and then you want to know which features are important. That's the most traditional, the definition of explainable AI. You want to measure the importance of features. Okay. Later, you will hear more about more contemporary, definition of explainable AI methods. Why is he saying, there's some error message? Okay, I removed it. Okay, so my lab has developed a series of, explainable AI methods. The first one being SHAP, which is a general framework, and it can be applied to any model. It doesn't really matter whether you're using deep neural network or ensemble trees. And then it has a desirable theoretical properties, which is one of the reasons that it's very widely used. We are not going to delve into this what theory it means. It basically means that it satisfies a set of axioms from it is called a fairness axiom, that very intuitive criteria which we want this methods in this domain to have, right? So when you get this feature importance measure, you better be it better be accurate, right? You don't want to use the method that's going to spit out any numbers in terms of features importance, right? So and so that using one of these versions, these developments, the tree SHAP, one version of a SHAP method, it is called the tree SHAP. With that, we revealed individualized risk factors for chronic kidney disease and also all-cause mortality. Will also delve into the problem of predicting clinical, the surgery duration based on various, patient data set. And then, these results, provided some new insights. So some of the results made sense, meaning that some of the important features, there was a consistency with the literatures, and we worked deeply with, especially for CKD, we worked with the UW nephrology department to make sure that its features make sense. And we also identified some new features that that could lead to new, scientific discovery. Okay, so that's what you can do with this SHAP or explainable AI method. So that was one example. And then just want to talk about, although it is an old paper, it's a 2018 paper, but it's a very first clinical AI work we have done in my lab. And then I just want to show you an example of how that helped clinicians' ability, how it helped clinicians to more, better predict a certain clinical outcome. Okay. We focused on a condition that's called hypoxemia, low blood oxygen level, that can sometimes happen to the patients who are connected to the ventilator because they are under anesthesia. Patients who are anesthesia, they don't breathe by themselves, and then that can cause various kinds of complications. This is one of them, and it's dangerous. And then our clinical collaborators, they really wanted a way to predict this in advance. Say, in the next 10 minutes, is hypoxemia going to happen or not? Okay. But not only that, they didn't like the idea of being just warned. They wanted to know why. That prediction was made by your complex AI model. Okay. So we developed a prescience system that can predict hypoxemia in the next 5 minutes, and then in real time and explain why. So what do I mean by explain? So it outputs the risk prediction in the order, in the unit of odds, the fault risk. So in this example, it's 2.4, some risk, not a lot of risk. And also its explanations, which show a set of features that increase the risk, that's a purple colored, and a set of risk that decrease the risk. Okay, so that's a green colored. And then, the plot at the bottom, this one, is equivalent to rotating this explanation bar at a certain time point by 90 degrees and stacking them horizontally. So you can see the time trend. So in this particular example, you can see that at the time, that current time point, the factor, the risk trend that, the large increase in the risk at the current time was driven by tidal volume, so meaning a drop in the patient's tidal volume of gas exhaled per breath. And then a patient, the anesthesia, as I said, they're connected to ventilator, and the tidal volume is one of the things that you can adjust in the ventilator. Of course, there is a separate question in terms of whether it's a causal relationship or not, right? But still, when you hear that, the hypoxemia is coming, in this case, hypoxemia actually happened, you can know, what reasons, what factors made that kind of, prediction generated by AI model, drove that kind of prediction. Okay. So importantly, we showed that the explainable prediction might improve anesthesiologist's ability to predict the event. And we used a user study using a web-based interface. And we know that, the reason being that why not clinical trial? Well, we can then, ensure that both prescience and then the 5 anesthesiologists we worked with, they are given exact same amount of information. They have exactly the same, set of features. Okay. So, and then we got this result of this ROC curve. So this is a common way to present accuracy of any AI or machine learning model. You basically want this curve to be as high as possible, okay? And then you can see that, if anesthesiologists anticipate 15% of the event, and then with the prescience aid, they can, anticipate 30% of the event. What that means is that, if you extrapolate all these, numbers, then 2.4 million more, additional episodes of hypoxemia are detected. Okay, so that was about near-term risk of adverse clinical conditions. So in the next project, we focused on a much long-term, lifetime prediction task. It's about how healthy you really are. In other words, your biological age, okay, as opposed to chronological age. So we developed in our recent paper, so it's this paper, Lancet Healthy Longevity, we developed a new clock. We named it Enable Clock, explainable biological age clock. And then the idea is that it estimates your biological age, but not only that, or it uses this kind of a feature, demographic, exam results from blood, urine test, and lifestyle based on questionnaires. And then we use UK Biobank data to train a reliable model with high accuracies. Then, not only that, it also gives you an idea of which features contributed the most to that kind of prediction. So to give you an idea, let's say there is a 5-year-old woman who turned out to be 72 in terms of the biological age. Then, these explanations that increase or decrease the biological age come with the unit exactly the same as the prediction. So prediction is in the unit of years, right? Because it's a biological age. And then that makes it, makes this kind of result super interpretable. So when I said in the, a few slides ago, introduced this SHAP method, when I said, desirable theoretical properties, this is one of them. When you have a feature importance scores like this, it's in the same unit as the predicted result, okay? So you can see, how each of these features increased or decreased the biological age in the unit of years, okay? So you can see, cancer diagnosis by doctor, obviously, right? And then, we found the consistency is important across many experiments. So it indicates kidney function. Okay? So we also, did this analysis in terms of different cause of mortality. Fortunately, the UK Biobank data came with the, cause of mortality. So that way you can also, compute the biological age in the context of a certain disease. Say, neoplasm, meaning, cancer-related deaths, or, circulatory diseases. So that can also facilitate some, validation analysis where we take this age acceleration as a phenotype, biological age minus chronological age, that's your age acceleration phenotype, and then you run, GWAS analysis to identify potential genetic hits. And then we identified, we do that for, this cause specific biological ages, and then did analysis in terms of, how those findings are consistent with the literatures. Okay, so all more detailed results are in this paper, the right side. Oh, and then, we also wanted to make sure that our biological age clock is good. And we compared with 2 other most well-known biological clock, one being BioAge, the other one being PhenoAge. And then we, did this test in terms of, how well each of them predicts a 5-year mortality in a separate held out data set, of course. And then, to just give you an idea what kind of information is used, so these are the features that are used by the full version of Enable Age Clock. So it's 800, it amounts to 800 features. And then, but you can, generate various kinds of, various versions of Enable Age Clock, depending on, what kind of information you use. You can use primarily, questionnaire features, which are relatively easier to obtain, or, some subset of these features. So we created these subsets and then compared with other biological age clocks, and then showed that, ours is more accurate in a statistically meaningful manner. And plus, we give you the reason. We give you the reasons in terms of, why biological age is a certain way. So it was a bit, we were a bit surprised when our paper was published. They put, they featured our paper in that issue, the December. Okay, so among the diseases, this is significantly affecting biological age. We cannot miss Alzheimer's disease. Top 10, it's, the only top 10 deadly diseases in the United States that doesn't have, long-term effective cure, right? Or, preventive measures, or, so a way to reverse or slow down the disease progression. This one sentence is I always, put in my NIA proposal, specific aims. First sentence is always fixed. So, and then, this one reason, it can be attributed to some extent to the fact that we have such a limited biological understanding in terms of especially early cellular pathways, not like, end stage pathologies like A beta and tau. We need a better understanding in terms of earlier cellular pathways that go wrong in the process. Okay, so that means that we need to understand molecularly, this disease better. So in our initial study in this line of a project, we first, my student who graduated from the computer science, first collected RNA sequencing data set from multiple AD cohorts, consisting of both AD and control, healthy tissues. And then, at that point, it was all bulk RNA-seq data. There was not really, single cell, it's not single cell data set. There was no, single cell data set available at that time. We are going to discuss, single cell data analysis a little later. And then, we modeled then this 6 Alzheimer's disease neuropathological phenotypes based on gene expression levels, 20,000 genes as input of this multi-output neural network model. It's not popped up yet, so it's not now popped up, right? So, and then the goal is not the accurate prediction here, right? Because, these are post-mortem brain, right? What's the point of predicting neuropathologies? The goal is to better understand the complex relationship between gene expression levels and then this, AD neuropathological phenotypes so that we understand the disease better and then disease drivers for potential target, therapeutic target identification. So again, but, there are many advantages of using this neural network. First, you don't really need to fully harmonize phenotypes. So some of the studies, such as, say MSVB or, ROSMAP data, they can miss some of these 6 neuropathological phenotypes. It doesn't really impact hugely in terms of how we train the neural network. It's modularized, right? So you simply just don't, train certain part of the network. You can still, train a neural network that has all these variables, even if you have some missingness. And then more importantly, you can also then capture complex nonlinear gene to phenotype relationships, which you can't, do with linear model or, univariate model. So again, but, this interpretability question. So how do you then make sense of your data? How do you even, interpret this complex deep neural network model? So, but we now have a SHAP. At this point, we had the SHAP method, and then we used, different methods to compute the importance of each of these genes in terms of for each of this, this AD neuropathological phenotype. That way, we identified some, a set of genes that are universally important across all, the AD neuropathological phenotypes. And then you can do that for, neuropathology specific manner too. But the bottom line is that we presented a lot of results. So one of the results we were excited about was that if you, focus on this, the genes that are universally important, we noticed that many of the genes are related to microglia immune response. And then to just give you an example, top 6, it's a very short list, right? And then, you see TREM2, and then you also see K and STERN, which are both, have been associated with microglia associated immune response. And then, these genes have been, implicated in genetic GWAS studies before. Okay, so the bottom line is that, we identified this pathway, and then, this it was also highly sex differential. So it can give us some of the answers in terms of what is the sex specificity in terms of, this Alzheimer's disease biology. Okay, so and then, I didn't put that in the slide, but, the really interesting piece of result, I actually mentioned it in the slide. So this one. So when we use this model as is, we didn't fine tune. We used, this post-mortem brain tissue, trained this model to learn the relationship between input and output. We took this model and then applied it to blood data, blood gene expression data set. Still, this predicted output, there was a very significant correlation between the predicted neuropathologies and actual disease labels. That was really interesting. And then also, we did this kind of test in a different species data also. So we got the gene expression data from brains, from mice, So this, cross species or cross tissue, it was from that. And especially blood, conservation is really exciting, right? Because that can lead to pre-symptomatic marker detection, because then you can use blood basically to see, to predict what will be the neuropathology level in the future, in the brain that will be accumulated. Okay, so that was interesting. And then, following that, we delved more into the optimal utilization of explainable AI techniques for gene expression data when they are input. Okay, so we are focusing on two significant enhancements. I was going to say, this student who led the project, so Joe Janizak, he is from the MSTP program, majoring in computer science, just got matched to Stanford. So I try to highlight, the lead authors when I give a presentation. As I said earlier, it was research is all done by the trainees, not really me. So anyway, so we incorporated two major changes. So first one being that, this current XAI, explainable AI, attempts to explain the prediction based on the input features. You go all the way to the input level. In this case, individual gene expression levels, which are not necessarily the most interpretable, right, compared to other functional units, such as biological pathways. So, and then also, sometimes unsupervised modeling can be beneficial, especially when you don't have a full set of phenotypes measured. And then, how does it work then? Unsupervised modeling means that you have original gene expression data, you do low dimensionality reduction, and the creating embedding space. So you have this low dimensional embedding so that you can reconstruct the original data set well. Okay, so that's the idea. And what does explainable AI means there? Well, I mean, you can then identify the genes. You still go back to the input feature level, and then you identify the genes that are important in explaining the expression variation within the data set. So you don't have a specific Y variable here, right? You don't have this prediction variable. Instead, you have a data set and a set of samples, and you are asking which genes were important in this general overall variation of the expression across these samples. Okay. So then, now Joe can go. So here is this architecture. It resulted in this new interpretable neural network architecture where in the embedding space, we organized this latent variables in the embedding space such that each latent variable represents a certain biological pathway, and it is connected to some specific set of genes, not all genes. So you can use various, the publicly available pathway databases such as CAG or Reactome. There are a few more widely used. So, and then, get a sense of the genes that are involved in the same pathway. So that way, in your embedding space will have a biological meaning. It will give you an idea of how active each biological pathway is then. You can still attribute back to all the way to genes. Well, first of all, you can get an idea in terms of which pathways are important within the data set, important in this, gene expression variation. Also, you can go all the way back to the gene level so that you can identify potential drivers for that pathway, okay? Master genes, master regulator in that pathway. So now we went back to this, AD data sets. So since we don't have to worry at all about the phenotype data, we could gather more samples. You know that, in deep learning world, the more samples are the better. So we got like twice more samples. And then, we identified oxidative phosphorylation to be really important. We delved into this, this latent space corresponding to that pathway, and then concluded that among the genes, this mitochondrial complex 1 could be really important in this data set. Again, remember this data set consists of both AD brain and then also healthy tissue. So what latent variable or pathways are the most important in this population of samples that have that kind of variation? It could be something that's, fundamental, latent factors that can be, interpreted as a disease-driving pathways. So that was mitochondrial complex 1, our computational hypothesis. You may wonder, how do you know, right? So we did a follow-up experimental confirmation in collaboration with Matt Cableland's lab, who have a wonderful animal model for Alzheimer's. So these worms produce A beta in their muscle at certain and then in certain age, in the certain point in their lifespan, they are going to be paralyzed. So you will visibly see when this A beta is going to have an impact on this, disease animal model. So, and then, we knock down each of these genes in this pathway, just a single gene. And then that even, led to meaningful delay in this paralysis time, every single gene within this complex 1 pathway. Okay, so that can mean that there can be some way to, more, it might be a promising path to delve into more, biological mechanistic functional studies. Later, we found the capsaicin is known to suppress this pathway. After publishing this paper, I am eating more peppers. So maybe a good idea. Okay, so then we try to come up with to tackle, analyze this a single cell data set, and then thought about, how to make it even more interpretable, our single cell data analysis. So we focused on the fact that single cell data sets are often collected to address contrastive questions. So what is the expression signatures that are specific to target? It could be diseased tissue compared to the background. It will be healthy tissue. It could be drug treated versus non-treated. It could be CRISPR, edited or not. Okay. So we often collect, single cell data set that way. So to better understand this heterogeneity, the differences, to really focus on the differences. So for example, disease process rather than the tissue specific normal, mechanisms, we came up with another interpretable neural network architecture that clearly separates shared variables from salient variables. And as you can see, for control samples, in this case, cells, because it's a single cell data, you just set them to be 0. Activation is 0. So that way, salient variables in this space, you are going to capture this low-dimensional embedding that's more specific, that's specific to this target samples, okay, target cells. So when we applied to the data, we named it contrastive VI. So I forgot to, 2 PhD students are co-first authors in this Nature method paper. And then we, where was I? Okay, so we applied this to the data from drug-treated cancer cells versus untreated cells. And then we see these cells behave differently in a meaningful manner in the salient space. Meaningful manner means, depending on their T53 genetic mutation status. This is what's known. It's already established, science. And then we confirmed that they behave differently depending on T53 genetic mutation status. And then this was possible only because this shared space absorbed this nuisance cell type specific signals. The single cell transcriptomics, it's mostly, that's the most dominant biological variation, right? And then it's absorbed all there because that's what's common between treated versus non-treated. So this kind of finding is obviously very important for personalized medicine. This is not our finding, by the way. This is what's already known, right? Yeah. So now we are continuing our research with the UW collaborator in pathology department, namely Jessica Young, to address the question of what drives Alzheimer's disease and related dementia, and then also if you think about this aging, you can think about a design where aged tissue versus young tissue, and then do the contrastive analysis. That way you can understand also, basic biology of aging better. You identify, expression signature and then potentially drivers. So that's also ongoing research. Okay. So now in the second part, which will be shorter, we will explore, how explainable AI can pave the way for brand new research direction. So you want to go beyond, just interpreting the model. Okay. So first example is AI auditing. So the first work, it was done by 2 MSTP students. And, just to give you the conclusion, in this study, we revealed that many, published clinical AI models to predict COVID-19 based on chest X-rays learned shortcuts rather than genuine pathology. And then our results, initial results, illustrated some potentially dangerous situation in which the algorithm appears to be highly accurate within one data set, even in the cross-validation setting, when you have explicit separation between training and testing. That's, red colored curve. And then, but fail when tested in hospitals external to the training data, that's green colored, okay? So we tried hard to understand why. And then we, took a look at the saliency map. So it's a form of explainable AI. You want to know which pixels are important. In this case, input pixels are features, and then explainable AI, traditional explainable AI techniques will tell you which input features were important for a particular prediction. Okay, so let's say that you have this chest X-ray image, which was predicted to be normal, COVID-19, negative COVID-19. It's perhaps right that the model used a clear long basis as a basis for determining negative, but it is not quite right that the model used the laterality marks, see like that kind of, this artifacts, or, the medical devices the patient was wearing. These are certainly not the reasons we want our model to rely on. Okay, then it will not generalize well. So we did many, kind of analysis based on this, this SHAP-based, the feature attribution methods. And then basically, stop the field from moving in a wrong direction. There were already 6 papers, very prestigious medical journals published, and then hundreds of related models, to be used out there. So X-ray-based, diagnosis, it can be really effective for low resource areas. We know PCR is the way to go, but and some of this model was going to be used in India. There was a startup. They highlighted the fact that they are using AI, and that we emailed the CEO saying, hey, did you know did you know that this model you are going to use, it's very likely it's one of the models we audited. So and they got a really nice reply saying they read our paper and they retrained the model. So clearly made a big impact. So after 1 year, this Nature Outlook posted a really nice perspective highlighting our paper. See, these are the 2 students, first author students in the rooftop of our building, and highlighted this work. So we now continue to try to dig into the flaws. It sounds really bad, but sometimes it is the performance of this models that are about to be used. So reasoning process of this clinical AI. Our next target is dermatology. We are working with Roxana Danishu at Stanford. So the idea is that we want to audit 5 AI models. So we picked 2 academic models. One of them is Nature, and then 2 commercial devices, cell phone apps. And then, this one competition winner. There was a whole competition, number 1 in this competition. Results were, not really promising. And then you can find more about this, what the prediction result was when you test in a separate data set. It's December Nature BME. But more importantly, I want to highlight the fact that saliency map did not work. The traditional definition of explainable AI did not work here. So you can see that, important features, it's just pointing to the entire lesion on the image, right? So this is one example of the skin image. It's a dermoscopic image, but you can also, our system, these systems, you can also use a clinical image, meaning, even cell phone, camera, you take a picture of the potential lesion, and then, this cell phone apps we audited, they are going to tell you basically whether that's melanoma or not, okay? So this one doesn't work because it's just, it's not really intuitive. It's not interpretable at all. So we invented a new way. We developed a new way to audit the system or making it explainable. The idea is that you generate counterfactual images. You modify your original image slightly just enough to cross the classification boundary. So say this was normal, benign, and then you modify, make minor modifications, small modifications so that it becomes, melanoma. That way, you can understand how this AI model was thinking, right? So in this case, this image, you see this blue region, blue-white veil. I learned a lot of dermatology terms from this project. So it usually, it indicates melanoma, okay? So we now know that this AI model used these features in the right way, right? So, and then we worked closely with the two board-certified dermatologists to systematically characterize the difference between normal and then benign and then malignant. So left benign, right side, malignant counterparts of the counterfactual images. So you can generate many pairs of these images, counterfactual images, per AI model you are auditing. So that way you can know how each of these visual morphological features, visual features are perceived by each of the 5 models we audited. Those are the names. And then also there is also notion of consistency of literature, or the way dermatologists think, right? Whether that makes sense or not. You can see, modern derm is completely doing wrong in terms of the use of darker pigmentation, in case of malignant, completely different way, right, from other models, and also not consistent with the literature. That way you can know why this model didn't work. Yeah, it didn't work well, right? Okay, so now I introduced the really, I got a chance to write a 1-page perspective article with Eric in terms of this clinical potential of counterfactual AI, where I introduced the more broader perspective in terms of how they can use. Okay, so another solution for us to make our model more interpretable, or auditing process more interpretable, is to use the concepts instead of input pixels. So see that you say that you have this dermatology image, okay, skin image, and then if you think about how dermatologists think, or even humans, we don't view this as a bunch of pixels. To us, pixels are not features. We think about whether there's asymmetricity or border type, coloration, and so on, right? So in some sense, taking this image as just a set of pictures may not be interpretable. You may want to get a sense of, this is semantically, interpret this image in terms of a semantically meaningful concept. So the question is how to automate that. When you have an image, you want to automatically, get a sense of, this is concept-based features, right? You can, instead of manual hand labeling. So we used this image text foundation model, which will appear, in a day or two, basically, in the Nature Medicine. We took a clip and then fine-tuned using 100,000 pairs of the skin image and the text caption collected from PubMed articles and then many dermatology textbooks, and then we fine-tuned. So the idea is the contrastive learning. At a high level, it means that you want your image to be close to the text if they are paired. You want them to be far from each other if they are not paired, okay? So that way you can bring image and text into one embedding space. So you have an image input, you have a text input, they are embedded into one space, and then you can get a sense of whether a certain image is close to some text prompts. So that can be a way to automatically annotate each image based on a set of meaningful or not meaningful dermatology concepts. We have even used, concepts like, is there, purple pen? That's, that's not really dermatology property, right? That's more like a noise, right? So, but we, you can still extract the concept level. Okay, so the next topic I want to briefly talk about is cost awareness of clinical AI. So many of the clinical AI, domain papers in that domain, people just celebrate, high accuracies and so on. But, if you think about certain areas like emergency medicine, accuracy alone is not the only aspect that's important. It needs to be cost effective. In this case, in emergency medicine situation, it'll be time, right? So to just, tell you more about this project. The leader was the Gabe Arion. He's MSTP, UW MSTP student who got PhD in computer science, now Harvard Medical School in emergency medicine. Really cool aspect was that this was his project, thesis project, published in Nature BME. And then, he got really into this, emergency medicine, and then chose this residency specialty. So what do we want to predict? So in this case, we are interested in ATC, very dangerous, condition. Let's say that you have a trauma patient. I said emergency medicine, right? You have trauma patient bleeding in front of you, and then you want to know whether this patient is going to have ATC, which requires is sometimes a massive blood transfusion, right? So if it's coming, you want to predict, of course, accurately, but you also want to predict fast, right? Time is an important factor here. So we work with a bunch of, the emergency medicine, physicians, led by Nathan White, co-senior author in this paper, and then a lot of, medical directors working with him to get a sense of how fast it is to obtain certain features. So we focus on nearly 50 features, and then got the idea of, approximately how many minutes or how many seconds it takes to obtain each feature. Train the model again with the, largely sampled data. But then we also use the explainable AI-based feature importance technique. So when you have a model, how much feature is important. Each feature is important. And then we did joint optimization in terms of that importance, and then also time cost, so that we have these two factors into account. Okay, we take two into account so that we can be accurate, but also with a low time budget. So in this result, it's not going to be ROC curve you may use to measure the performance. It'll be more like, x-axis will be more their budget, how many minutes in total you would allow, and they will determine how many features you will use. And then y-axis is predictive accuracy, area under the ROC curve. We showed that co-AI is better than existing clinical score, that's a PACT score, that's a logistic regression-based method that uses, I think, like six features and takes 8 minutes. And co-AI, you can see that it's less than 1 minute, right? So that can make difference. So as I said, many of our methods are not domain specific, it's general. So we applied it to mortality prediction task using mimic data. So this is the field where there are a lot of clinical scores. So especially, QSOFA was specifically designed to address this, cost issue. And then you can see that, co-AI does better than any of them, the existing clinical scores. So the last example I want to show you is related to cancer therapy design, okay? Combination therapy. We focused on AML, acute myeloid leukemia. And then, choosing optimal combination is important, but it's also hard, because when there are hundreds of FDA approved individual drugs, there will be tens of thousands of combinations of these drugs, right? If you consider 3 drug combination, it will be even more. So you better know, why or why not any 2 drugs are going to have a synergy or not for a particular patient who will have his or her own, genetic characteristics, right? So we first developed express model that takes gene expression data. You get the blood or bone marrow, and then measure gene expression levels. And then for any 2 drug, A and B, we collected the drug-specific features, such as molecular structure or target pathways or target genes. And then use a complex machine learning model, high performance. You want the prediction to be accurate. But you don't stop there, you interpret, you also can know which genes were important for patient and then drug combination specific manner. Okay, so if this is the end of the story, this project would have been presented in the first part, right? So what are we, really interested in knowing here? Here, we focused on identifying the underlying principles. So for many drug pairs that have a synergy, what are the underlying principles and what are the rules, instead of delving into individual prediction for a patient and then specific pairs of drugs. So, and then, we did also the pathway level analysis in terms of, if some genes, set of genes are important and then they are enriched for a certain pathway, that can be important information, right? So the conclusion there was that we combined all these SHAP values for all drug combinations and all patients, and then to get a sense of what biological pathways are important in general. What would be the underlying really important pathways that determine synergy or non-synergy between any two chemotherapy drugs? So we identified this hematopoietic cell lineage pathway being one of the most important, and then we did many similar analysis in terms of which pathways are important, and then concluded that this stemness feature, that the genes that indicate how differentiated hematopoietic stem cell like signature, depending on who you talk in the cancer biology field, some people will use this term stemness, some people hate this term. So and then for shortness, I would say stemness turned out to be really important, and we can even consider that as axis. If drug A is known to work well for less differentiated cancer, you want the B to be the one that's known to work well for more differentiated cancer. Okay, so there was one of the many conclusions from this paper. Again, short introduction per project, so you can learn more about this project in our paper. So I presented, this idea of applying explainable AI in a bunch of examples. So the motivation behind it being that, to induce more collaborations, right? So to give you an idea of a broader spectrum where explainable AI can actually make an effect. I do want to say that this computing feature attributions itself is really difficult, challenging computational problem. See, as computer science, machine learning problem, we are actively publishing papers in this domain. And then we applied to wide range of problems in first medicine and healthcare. You can see the broad coverage of these departments and divisions of the collaborators we worked with. And then cancer biology and precision medicine, and also Alzheimer's disease, where there's no drug at all, and the biological understanding is incredibly important. Okay, so and then you can what I talked about is a small portion of our research in my lab. You can get to know better about our research by navigating through our publication website. One tip, though, is that this is a frequently asked question: what exactly is your lab working on? Because we work on many different topics. So I made some really nice labeling system implemented when I said ABC in terms of what we do in my lab. So I labeled every single paper, hand labeling, not automatic foundation model based, in terms of A, B, and C. And then also journal and conference. I recently added a perspective article too. Okay, so again, it was all done by my students, amazing students, and then I didn't write any piece of code or anything. It was all done by my students. And then now I would like to spend a couple more minutes to talk about what next. Where are we headed? Where should these AI researchers be headed? So before talking about far future, I just want to talk about the prediction I made a little more than a year ago. GeekWire reporter asked me at the end of 2022 in terms of what's coming in 2023. So it's already passed, right? So what will happen in digital health? And then I didn't hesitate to say we will start seeing XAI functionality in devices, FDA approved devices, and FDA will even use XAI to decide approval or not. And then I wouldn't assert that I was right, but I think I want to say that we are heading in the right direction. And then now it's not just about medical AI. Now a lot of people have some concerns about AI and they really want to understand how they work. And then but to be able to really learn whether it's a failure mode or a success mode, or even audit or regulate AI, you first need to understand what they are doing. So it would be the first step would be explainable these AI systems. So our future research, it involves just the bottom line is that there are so many things to explain. So and then we are focusing on both B and C. we noticed that underlying principles in terms of how AI or computer science in general can advance this area, it's often the case that underlying principles are shared. Sometimes the idea we used for the project that's under the B category, it can be applied to some of the problems in the C category. Currently, we see two separate fields, right? Computational molecular biology or systems biology versus clinical informatics. Totally different, communities and conferences and journals. But, the future of medicine requires the convergence of them. Medical decisions need to be made based on, really true understanding, biological understanding of the patient. So we are continue to developing concept-based explanation methods. The Nature Medicine paper I told you a minute ago, it's only one example. And then also your explainable AI technique for multimodal foundation models. You really need to understand, what they are doing. Foundation models, there's a lot of promises, but also concerns too. So more, auditing, more model or data auditing, and then also, something for generative AI as well. Okay, thank you so much. And then I'm now happy to answer any questions you might have.
Questions from the room for our speaker? And if you could just repeat them for the folks
I
on
will.
Zoom as well.
Yeah.
Good. Go ahead.
Yes, please.
Thank you very much. I had a couple of questions regarding SHAP. So first of all, is the SHAP a measure specific to each feature? Can you also be specific to each feature for each image or patient, or is it individual?
Yes, individual specific. We call it local feature attribution method, in the explainable AI field, versus global means global importance across samples. So SHAP is going to give you the local answer. Yeah.
Basically, everything so far additionally subdivided.
Oh yeah. That's the whole point. Yes. Individualize the risk factors in some of the examples.
And the other question I had was, I think you presented this was on Alzheimer's disease. Your model was trying to predict different phenotypes of Alzheimer's disease given gene expression. Did you try to check whether there was correlation between the SHAP and the genes across these regions?
Yeah. So our main results involved universal importance. So the genes that are universally important across these phenotypes. But I get your question a lot actually in previous talks. So it will be really interesting also to delve into the difference between, say, like A beta and tau. What are the differences in terms of the important genes, right? Yeah, so that can be really our supplementary material contains all SHAP values for all samples, so it can be a good way to. But in our main paper, we focus on universal importance first.
I wonder if there may be like local correlation specific.
Yeah, that might be really interesting. Yeah, I even think that differential SHAP can be something new, interesting method we could develop. Yeah, Trey.
Great talk. Here we go. I wanted to ask about the time-aware predictions near the end of the last third of your talk for the ER predictions, or EHR.
It was.
So the model itself runs instantaneously. It's pre-trained. So to press enter on a patient's data is instantaneous. So when you're talking about minutes for a prediction, is that actually gathering? It's actually gathering the data from the patient.
Right.
And some features take longer to gather than others. Is that how to understand that part?
What we did is, we can optimize in many ways in the future project. The way we did was the plus, the feature time cost of feature number 1 plus that of feature number 2 plus just aggregate.
So is the model making a prediction off of the first feature and then it has
No, not that.
So is there an active learning aspect to this? I guess what I'm trying to
It is active learning.
and could there be, so it is active learning.
Yeah.
Great.
Yeah, we were asked to actually, compare with the existing active learning methods. Yeah, during the review process, we did so.
Yeah, nice talk. I wonder for the AI, do you consider to add a dog domain? What I mean is like that. Actually, that I was watching YouTube quite a few times, and one of the patient, actually, one of the dog owner is actually a physician, and he's a cancer, a physician, actually, oncology. So the dog kept licking one side of his face, and end up to be is a melanoma. So and I was wondering, because actually dog have a, if you go to YouTube, dog is actually expert in spelling colon cancer, breast cancer, and melanoma. So that would be a plus for the AI, because they call that a dog factor.
So I didn't get your question. So so I didn't understand your question. Can you
use the dog to train the AI?
using what?
Dogs,
Doctors,
dogs.
dogs for
Same for your training examples. Dogs are better than doctors at generating training data.
Oh my God. Okay.
Time for one more question. Out of competition here.
Thank you for awesome talk. I have a question. If the AI model is calibrated and how we can interpret sharp score, is it like a contribution, probabilistic contribution to calibrated output, or like a,
What
we
is
need to
it,
build-
right? Yeah.
Yeah.
What is the theoretical basis for that? Is that your question? Yeah.
Yeah, so sharp is like.
I know what you're wondering about. So I may have a hidden slide. Let me take a look. So how does a sharp work, basically, right? It's not really probability. Yeah. How about I just give you like 1 minute? Because sometimes I have that when the computer science, is the main audience. And then, so to give you a very brief idea of how SHAP works. So what is the key question? Key question is whether how much feature I is important, right? So, and then reasonable way to address that question is you take out I, right? Yeah, or you have some existing set of features, you add I, and then measure the change in the Y in that model, right? Change model output. But then it may, it may depend in terms of what other bodies feature I will have, right? So you have some subset of features, you add I, you want to measure the model output, but let's say that subset, you consider all possible subsets and then take the average. Sounds very reasonable, right? Yeah. So that's the sharp values. That means a sharp value. And then game theory, in the old game theory, gives you many theoretical properties. If you do particularly that way, it satisfies many of the desirable properties to be this feature attribution method. So the bottom line, so that's why you see a lot of Shapley value-based approaches, because there is not a good way to compute accurately, because you have to always approximate. Unless you are using tree model, using our method, tree SHAP method. So, the idea to just go back to your question, what it really means is that how much feature I, it's not probability, how much feature I is gonna change the model output, average effect, depending on what other bodies are. Yeah, so that's the definition of SHAP value.
Thank you so much.
Mathematically.
Thank you, speaker.
Thank you very much.